Hello everyone, welcome back to another session of regeneration biology. In today's class, we will learn about major challenges in tissue engineering when you apply it as a practice. So this class may look little bit technical and we will try to take them as light as possible, but understand this is also very much important when you try to understand how you apply tissue engineering and organ culture etc into practice, simply because in reality when you try to implement something, the problems you face may have nothing to do with the actual science. For example, in reality you can make a ideal footwear which is very comfortable, very smooth, friendly to your feet, everything. But when you try to manufacture it, if the manufacturing cost is becoming 1 lakh rupees, then we know it is not going to be sustainable. Or in other words, it is cheap. You are trying to implement it for walking on the road. Then if it is not made of the right material, it is cheap, but it will not stand the rough terrain on the floor. So simple example I gave, shoes. So what you have to understand is cost has to be taken care of, ruggedness has to be taken care of, friendliness to the feet also has to be taken care of. So tissue engineering also is something like that. So let us see some of the challenges. One, this is basically a picture that is showing how the tissue engineering setup is arranged. So one thing, immediate point to remember is the microenvironment. The proper reconstitution of the microenvironment for the development of the basic tissue function and properties is a important point to consider. Cell communicates through the local microenvironment, and if the environment is not friendly, like you can see in this model, there is 100 micrometer tissue microenvironment. So we should know 1000 micron or 1000 micrometer is 1 millimeter. So 100 micrometer means 1/10th of a millimeter. So there are many cells are there, progenitor cells, hematopoietic accessory cells, etc. They are all adhered onto a substratum. Let it be natural or artificial, doesn't matter, decellularized or artificial, doesn't matter, it has got a matrix. And this has to be put into a bioreactor, and this bioreactor now is assembled into multiple pieces into around 10 to 100 centimeter automated cell culture system, and these automated cell culture system will be clubbed into 1 to 10 meter in height, length and width, and it's a cell manufacturing facility where you will house them, means the size of large rooms. So this is a typical way of arranging. And then the neighboring cells, what are the cells that is present? Like we have seen, when you culture the stem cells, they need to have a feeder layer, etc. The interaction with the ECM, signaling molecules, cell geometry, dynamics of respiration, supply of nutrients and removal of metabolic waste products, etc. Everything has an implication in posing as a threat to the cell culture. And it mimics this microenvironment, it mimics the dynamic chemical and geometric variables that is normally seen in the actual tissue of the organism. Now the question is, once you set up the microenvironment, now how nicely we can scale it up? We have to generate numerically enough, or it should be viable enough or scalable enough. Properly functioning microenvironment has to be established. Say for example, you have got one group of a small bioreactor, and now you assembled into 100. Now it can't happen that 3 of them are not receiving the nutrition, then whatever organ you are making can be futile. So each unit has to have its complete responsible distribution of nutrients, and then only you will end up getting a clinically meaningful tissue outcome. And then comes the microcirculation. How are you circulating the nutrition, which has got the glucose, amino acids, oxygen, etc. It connects the microenvironments to every tissue. And metabolically active cells are located within few hundred micrometers from the capillary which provides the nutrition. And capillaries connect every cells to a source and a sink. There is an input of nutrition and an exit of nutrition. And then the challenge comes on the automation of the system. How nicely you are automating it. In the sense, it should be viable. It cannot be like I gave an example. It cannot like you don't want a shoe to be made for an ordinary use of 1 lakh rupees. Same way, automation system should have its own cost effectiveness, and so that it can work operationally, clinically, meaningful scale, it can be pushed into. So these are all some challenges, and let us see some other challenges when you are looking into printing an organ or making an organ. So different challenges and possible solutions. Let us see. Due to the complex biological properties and 3D ultrastructure of the native ECM, we have seen in the previous class how decellularization is important to get the 3D structure of the native ECM. Same way, 3D structure of the native ECM is as important as the organ culture itself. It has to replicate or mimic the using traditional manufacturing methods to that of like you are making a matrix artificially, which mimics the endogenous or the normal ECM of a organ. So this is the question. So traditional manufacturing methods and biomaterials can become often challenging. Like ideally, everything is working out, but when you put into practice, you find it, it is not strong enough, or it is not. It's just like if you are making a car also, you want the car to be very light, then so that you will be fuel efficient, cost effectiveness will be there. But nobody makes a car out of plastic. It will not be strong enough, very light, but it will not, by the time you close the door couple of times, it will break apart. You don't want such a car just because you want to reduce the weight. So there is a trade-off works behind this. In the cultivation and application of organoids, a critical challenge lies in developing mature organoids that possesses the functional and physiological properties of the native organs, essential for tissue repair and biological function reconstruction. Say you want to have extra one room, like you want to give a patch to your existing house, but you forgot to put roof, then that room is useless. Or you forgot to put the floor. Forgot in the sense it did not work out. Although you tried making it, it did not work out. But still all the walls and roof is there, but the floor is not there. So everything makes a difference when it comes to organ. So every organoid you are making must be a replica, miniature replica of the organ itself. To minimize the batch-to-batch variations due to different donors, some researchers are focusing on using stable cell lines to produce these extracellular matrix, thereby offering new insight into these challenges. Like you are telling the cells to release the matrix protein so that it will be dense enough or quantitatively and qualitatively enough to mimic a endogenous organ. That is some approach. So we have heard about the DECM, that is decellularized ECM materials, and let us see how it is working out for renal organoids in and engineered organs. Some examples we will see so that we will know more about the challenges. Renal means it is about the urinary tract or kidney, or based on which organism you are talking about or which part of the excretory system is affected. There is production of, or there is possibility of transplanting those organs. So kidney transplantation is an effective treatment for end stage kidney diseases, but faces a chronic shortage of kidney donors, which we know, which is a challenge in every organ demanding patients. Kidney organoids represent a potential tool for producing transplantable kidneys. The decellularization and recellularization of kidneys into kidney organoids provide a solid foundation to start with. Kidney DECM means decellularized extracellular matrix preserves the complex structure, composition and microenvironment of natural kidney tissue, supporting the self-organization and differentiation of stem cells for progenitor cells into specific kidney cell types such as nephrons, collecting ducts, stromal cells, closely resembling the cellular diversity found in natural kidneys. This is what is demanded. But are we getting in the reality? That is what we should focus on, especially considering the challenges. Different decellularization protocols can yield materials with varying histological properties, human cell endothelialization degrees, and varying degrees of preserved microvascular integrity and functionality, thereby influencing the differentiation of various embryonic stem cells into renal lineages. This is what is implementable thing. This offers new insights into the importance of optimizing decellularization protocols. Means your organoid is only as good as the DECM you started with. So looking further into the kidney organoids, kidney transplantation is an effective treatment. We know that kidney organoids represents the potential tool. The decellularization and recellularization must be done effectively, and the kidney DECM has the ability. It can be taken from a pig, it can be taken from any other related organism. All you care is the 3D structure. And now you should put this DECM into a chamber, which we call it as a bioreactor, where you will seed the DECM with the cells, and the cells which you are putting will be closely related to the patient or from the patient himself or herself. But now the question is, these cells should accept that DECM as its own native matrix. If it did not, for some reason it rejected, then they will not colonize. So this also has posed several challenges. So let us see the application of DECM materials in renal organoids in engineered organ culture. So this is a schematic illustration of the conventional and DECM derived bio-inspired scaffold for renal tissue regeneration, which highlights their biological and chemical mechanism. You can see here, and in the panel A and panel B, which is basically you are using a scaffold. It can be artificial, it can be natural, but in either case you need to seed them with the cells. And in panel C, what you are seeing here is that they get into once you seed the cells, they undergo inflammation, and you will end up getting a if it is not proper scaffold, you can get too much of fibrosis. If it is the right way of ECM performance, then you end up getting proper tissue that will be generated. And different like here it is, what is used is PLGA, polylactic co-glycolic acid. That is the scaffold that is used. Different scaffolds, chemical nature is being used. We will not go behind them as of now. And in panel C, the decellularization and gelation processes of porcine kidney. Porcine means pig kidney. They have taken removed kidney from the host, and then they get rid of, decellularize it, and they lyophilize it, kill all the cells just to get the DECM, and which is ready for colonization of the host derived cell. And then you can see in the panel D is the characterization of these cells, whatever they got it, whether they are good, bad, are they colonizing, etc. from this DECM. And renal organoids cultured on kidney DECM, they exhibit more mature tubular structures. Tubular structure means that internal anatomy of that kidney has to be restored back. Otherwise, you cannot make urine out of, or that kidney will not be able to filter urine out of it. And let us see a few more challenges in this approach. Kidney DECM is one of the most promising biomaterials for constructing renal organoids and bioartificial kidneys. However, insufficient recellularization of scaffolds, like I already told you, scaffold is fine, decellularization is fine, but the inoculated cell or seeded cell is not finding it a comfortable place. So this inefficient recellularization can be a problem into the scaffold in vivo, still challenges the application of kidney DECM. Sometimes it can happen that it is not anything wrong with the DECM itself. It is something wrong with the cell what you have seeded. If the kidney of the patient was perfect, or this organ's body was perfect, then their kidney would not have gone bad. So what you are trying to mimic is not so optimum or not so perfect cell. This also can pose a challenge, but can we call it as a challenge is the question, because we cannot tell a patient that your organ is failure, so if it is successful, you will get another life, otherwise I don't care about your life. That is not possible. Even though the cell is bad, we should be able to fix it. Then only it becomes the right approach. Generating complex organ like kidney through 3D bioprinting presents several other challenges also. First challenge, the kidney structure is complex. This we know. Composed of macrostructures such as renal arteries, veins, ureters. Every organ has to have arteries. We discussed about angiogenesis already, because without angiogenesis, that organ is waste. At least it will not stay in the body of the patient. But kidney has got much more complex internal structures. They have got renal arteries, veins, ureters, and other microstructures like nephrons and the stroma that occupy the space between them. Everything has to be perfect, because kidney urine filtration depends on the proximity, intracellular and extracellular, and the movement of ions, so that there will be a concentration gradient will be created, so that the lost ions will be absorbed and the lost fluid will be absorbed. Lots of functions it does, everything based on its structure. Secondly, the selecting the correctly positioning of the cells. Correct positioning of the cells is challenging, as the kidney has over 20 different cell types. Although it's a small organ, it has got 20 different cell types are there. Not that you have 20 cell types put randomly. They have to be arranged in the place where it should be. Moreover, choosing suitable biomaterials to preserve the printed kidney structure and function warrants careful consideration because of the future immune rejection, etc. Let us see few more challenges. In recent years, progress has also been made in kidney fabrication via bioprinting. So combining 3D bioprinting with the renal organoid technology allows for the rapid high throughput generation of renal organoids with reproducible cell numbers and cell type ratios. That means the printing allows you that this cell should be here, and this, because you are already making a 3D scan of the organ and you are printing precisely, just like you are making a color print. So you have a blue flower and red color another flower and green leaves. It will never happen that red color flower now looking green in color and the blue color is looking in white color. It never happens. It will be blue, green will be green, red will be red in that particular place, not randomly somewhere. The same logic applies. However, generating complex organs has to depend on the 3D bioprinting. The choosing the suitable biomaterials also is used to preserve the printed kidney structure and function, and which is essential for maintaining its future. Otherwise, you can make a kidney now, but it should be standing in the or staying in the patient. DECM materials for hepatic organoids and engineered organs. Another organ which is very vital. The liver is a vital organ for metabolism and homeostasis in the body, which we all know. The high mortality rate associated with end-stage liver disease, ESLD, it is called. It has posed a significant global public health challenge, because liver is one of the most affected organ or the most hardworking organ, we can say, in your body, because it is trying to safeguard you every day, because every food what you consume can have some or the other toxin. So liver's job is to detoxify it. Data from the World Health Organization show that liver diseases ranks as the 12th most common cause of death worldwide. That is a large number, considering it is not heart, it's not brain, etc. Still, liver damage is more prevalent. That is what you should understand. Not only just prevalent, it can lead to death as well. Currently, orthopedic liver transplantation, known as OLT, is the most effective therapeutic option to enhance the survival and life quality of ESLD patients, that is end stage liver disease patients. Whole organ bioengineering and regenerative medicine represent promising new technologies that could alleviate liver shortage by increasing the number of organs available for transplantation. Means the organ available for transplantation is much low than the people who are waiting in queue. So DECM materials are used to make hepatic organoids, as you can see in the schematic. Application of DECM materials in hepatic organoids and engineered organ culture. In panel A, you can see the preparation process of DLS, how you make a native liver and decellularize it. So this is the liver with the blood, tissue, everything. And finally, you end up getting a matrix, decellularized matrix. The morphology of their intrahepatic bile duct tree, that is what you see in the B. And in panel C, the vascularization process of the DLS. What you are seeing here is the vascularization process. And in D, microscopic images, as you can see here in panel D, there my ultrastructural images of the matrix. And E, scanning electron microscope images, SEM images of DLS, that matrix what you created, and the vascularized liver scaffold VLS. Like you can see A, B, C, and D. They are the vascularized images, as you can see here. And in F, panel F is a photograph of the perfusion culture apparatus placed in the CO2 incubator utilized for recellularization of bile ducts in DLS. So how can you make a proper bile duct? Because bile duct defect also is a very serious, it's as good as no liver functioning. If your bile duct is damaged, it's one of the complex structure in your body. If that is damaged, it's as good as liver not functioning. So then G, panel G, reconstruction of biliary tree like structures that was achieved through the recellularization of bile ducts using liver ductal organoids. And this is the microscopic images of their functionality. Those who are interested can read this article. So what are the other challenges and limitations of the 3D printing? Although we are saying, okay, 3D printing can be done to solve the problems of shortage of organ, 3D bioprinting has several other limitations. Has a potential to revolutionize tissue engineering. T stands for tissue engineering and regeneration, but it plays an important role in personalized medicine by enabling the design, prototyping, and fabrication of 3D tissue structures for various therapies. Means you can make, like I gave you an example, you can make a person-specific shoes, person-specific dress, like that you can make a person-specific organ because of the 3D printing. Although the 3D printing has progressed considerably, the entire biofabrication platform must be standardized and integrated from software design to tissue processing after printing. It's like I told you earlier also, designing or conceiving an idea is one thing, implementing in reality is another thing. So both many times doesn't go well. So we use the software designing for this 3D printing approach. To develop next generation bioengineered tissues, it is essential to address the drawbacks of biofabrication platforms regarding speed, complexity, material selection, printing resolution, and cell processing. This has to be integrated effectively. The choice of bio ink. So far we have been talking about printing, but what will be the ink used? So we use the terminology bio ink is one of the most important aspects of bioprinting, and it helps to circumvent some of these challenges. We will see more about these things in the bioprinting area, but time being, get familiarized with the terminology bio ink. You use something called ink, bio ink. Bio inks mimic the complex structure and composition of different organs and tissues. Means you are printing organ, so the ink should be so good in quality that after printing the organ should look like an organ. You don't want an organ to look like a wooden piece or a plastic piece. They act as a medium to protect cells during the printing process and provide a suitable environment for developing microtissues to mature following printing. So this printing should happen, the cells will find in a printed organ a familiar place to live. So the bio ink should have a viscosity suitable for cell growth and differentiation, as well as printing. However, when you try to apply in practical, it should have the suitable viscosity for bioprinting may not support cell viability. Like again, I gave an example in the beginning of the class. When you make a shoes, cost is one thing, and here what you are telling, the viscosity when you use for bioprinting, maybe it is ideal for printing, but it is not ideal for the cells to live in. So then you try to make it ideal for cells to grow, then it is not suitable for printing. So this is the trade-off what one has to take focus on to. On the other hand, shear stress can affect cell viability and bio ink's stability. Once you make everything perfect, then ink will not print. It will not be able to. So you should think about the strategizing how the ink will suit for the cell's familiarity. Another consideration is cellular sources for constructing 3D bioprinted tissues. Primary cells are mostly pre-harvested in vitro before heterogeneous tissue constructs are bioprinted. Primary cells for transplantation are removed from the patient to avoid any immune reaction. So this is the approach that is done usually for any bioprinting approaches. So what we should try to understand now in conclusion is that the DECM based organ culture is designed to remove immunogenic cells while preserving the original tissue architecture and composition. Means like you are harvesting, say you are harvesting something from the farm, you are taking, say, take about the wheat or rice, you are harvesting the whole plant, but then you are getting rid of the hay, etc. Something like that. You are making use of every beneficial thing, but you are getting rid of those parts which can be immunogenic or not so friendly for the recellularizing cellular components. Due to its inherent structure, enhanced bioactivity, reduced immunogenicity, a favorable biodegradability, the DECM has garnered widespread attention in tissue engineering and biomedical applications. While the 3D printing remains an active option, the decellularized extracellular matrix and recellularization of them always remained as an attractive proposition for tissue engineers. Despite the challenges in exploring DECM for organoids and engineered organ fabrication, ongoing research and technological advancements bring us closer to creating viable transplantable tissues and organs. We are reaching step by step towards a very sustainable and suitable approach, whether you should go for an organoid production via 3D printing or production of an organoid production via decellularized matrix. Everything depends on what is the approach which you are going to take for fixing a damaged part of a patient. These developments also include optimizing decellularization process, that also matters, decellularization process, refinement of organoid construction methods, scale-up production, and enhancing the matrix biological cues to guide cell behavior. Another approach. So the continuous improvements of these technologies holds great promise for the future of organoid engineering and engineered organ fabrication. So we should understand these technologies has to be implemented wisely and effectively in a patient dependent manner. Then it will become a very suitable approach for humanity. We will learn more about 3D printing and organ culture in the next class. Thank you.